morning, dear students. Today we are going to start our class with the topic mode of infection and pathogenicity of Leishmania. What about the hierarchy of this Leishmania? It comes under the domain Eukaryota, phylum Euglenosova, class Kinetoplastia, order Trypanosomatida, and genus Leishmania. And the flagellates genus Leishmania, it is morphologically similar or identical to those flagellates coming under the genus Leptomonas. They both, Leishmania and genus Leptomonas, they both come under the order Trypanosomatida. So, as we look into these pictures, we can see that morphologically they are somewhat similar. Then, which are the clinical syndromes, diseases occurs because of Leishmania infections? They are of three: visceral leishmaniasis, mucocutaneous leishmaniasis, cutaneous leishmaniasis. Then, which are the hosts of Leishmania? Human and insect hosts are there. Insect specifically sandfly. They are usually the vectors of this parasite. Its picture is shown here. And there are two morphological forms for this Leishmania. They are promasticoid form and amasticoid form. Its pictures are shown here. This is promasticoid form and this is amasticoid form. This promasticoid form is found in this vector fly, usually in this vector fly, sandfly's body. And amasticoid form, this form is found in the vertebrate host like human. Sorry, amasticoid. Then case history. If we check the case history of leishmaniasis, then we can understand that throughout the world, there are about 12 million cases of leishmaniasis. And in every year, it is estimated, how many cases estimated to occur? 1.5 to 2 million new cases every year. So that must be its severity. Then regarding the geographical distribution, we know there are 7 continents and among them, except Antarctica and Australia, all the other continents have the infections because of this leishmania. And in these countries like India, Brazil, Sudan, South Sudan, Somalia and Ethiopia, in these countries, among the reported cases, 90% are of new cases according to WHO. Okay, then this Leishmania donovani, it causes the visceral leishmaniasis and it is prevalent in India until 1947. Now, it is somewhat sporadic. Sporadic means it is not that much common. This visceral leishmaniasis now also it is sporadic with a few cases from Kerala and Gujarat. Then we need to understand about different groups, different forms in leishmaniasis. We can classify the leishmaniasis into different groups. How we can classify? That is according to its development inside the vector. Vector means phlebotomous sandfly. In that fly, how? What about its development? Based on that, we can group. Promasticoid form. It develops in the digestive system of the sandfly. So, promasticoid form is developing in the digestive system of the sandfly. And the names of the grouping, it is giving in accordance with its relative position in pylorus, in stomach. Which are the different groups? Saprofilaria, perifilaria, and hypofilaria. How they differ? First, let's see what is saprofilaria. Saprofilaria, this one is the most common variety that is affecting leishmaniasis. Saprofilaria forms. This is the most common form responsible for leishmaniasis. And its development takes place in the pharynx and stomach of sandfly. Okay, its development occurs in the pharynx and stomach of sandfly. Then next one is perifilaria. Perifilaria, its responsibility in causing leishmaniasis that comes next to saprofilaria. Yeah, and its development occurs both in pharynx and stomach, and also in the anterior ileum. Ileum, we know it is the last portion of small intestine. Three regions its development occurs: pharynx, stomach, and anterior portion of ileum of the sandfly. So according to that, we are classifying all these varieties. Last one, hypofilaria, and its development occurs in the ileum and rectal region. Rectum, we know, it is a part of large intestine. Then which are the main forms of leishmaniasis? Which are the different types of leishmaniasis disease? Three main forms exist. First one, visceral leishmaniasis or kala ulcer or black sickness. What is the feature? What is the peculiarity appearance of this disease? The color of the skin turns black. A black pigmentation will appear. We'll show its picture. A black pigmentation will appear in the skin. Then this parasite usually infect the internal organs. That's why this disease is called visceral. So as it affects the internal organs, this leishmaniasis is called visceral leishmaniasis. And sometimes it will not affect internal internal organs that much, but it causes some lesions in the skin. So that is visceral leishmaniasis. Next, visceral leishmaniasis itself we can classify. We can categorize into three different types on biological basis. That means those parasites that found human as the reservoir. Those parasites is causing the Indian gala ulcer. Then canine reservoir means those parasites that found canines as the reservoir, they are responsible for causing Russian gala ulcer, then infantile or Mediterranean gala ulcer, Chinese and South American gala ulcer. Then in rodents, some parasites found rodents as the reservoir, they are responsible to cause African gala ulcer. Next leishmaniasis type is dermal cutaneous leishmaniasis or oriental sore. And the causative species is Leishmania major and Leishmania tropica. And this parasite is distributed in the subcutaneous tissues, means skin, and that infection will cause local lesions in the skin. You can just see a picture of that. Lesions. Local lesions is possible to see. Then dermal leishmaniasis itself we can categorize into two based on the type of the parasite. Again, like visceral leishmaniasis. Leishmania tropica means it is a species that cause oriental sores. Leishmania donovani, it causes certain non-ulcerative skin lesions, and these lesions are usually produced after kala ulcer, dermal leishmaniasis or dermal leishmaniasis as a last phase of this type of infection. This leishmania donovani cause non-ulcerative skin lesions. Next is mucocutaneous leishmaniasis, and its causative species is Leishmania mexicana and Leishmania brasiliensis. And this parasite, it affects the mucosal lining of oral and nasal portion and cause local skin lesions. And we can just see the picture of the infection. It is very disgusting to see its appearance. Okay. We are going to learn about the type Leishmania donovani. It is a digenetic endoparasite which causes kala ulcer in human. This primary host is human and the secondary host is sandfly. Then let's know something more about that sandfly, that is phlebotomine sandfly. It is a fly that is active from dusk to dawn, and it is 2 to 3 millimeter long. And how that infection is transmitting that fly to a human by the bite of an infected female vector to human. By that way, the disease is spreading to a human. Then we are going to see the different morphological forms of leishmania. Two morphological forms, hemasticoid form and promasticoid form, exist. Hemasticoid form is otherwise called the leishmanial form, and promasticoid form is otherwise called leptomonad form. First, we can learn about hemasticoid form. Where it occur? It resides in the reticuloendothelial cells. What is this reticuloendothelial cells? Reticuloendothelial cells, they are a system of cells. It originated from monocytes and are able to perform phagocytosis of foreign material. Such a system of cells is called reticuloendothelial. So this hemasticoid stage, it resides inside the reticular endothelial system of the infected human. And what about its size? Its length is about 2 to 5 micrometer and breadth is 1 to 2.5 micrometer. Its picture is shown here. What are the features? It possesses a centrally placed nucleus. Then a mitochondria is there, and Golgi apparatus also there. Golgi apparatus we can see here. And also it possesses kinetoplast. Kinetoplast means the concentrated mitochondrial DNA also present here. And flagellum. At the base of the flagellum, that cell membrane invaginate to form a vase-like structure. What is the importance of this structure? It is the communication site between the parasite's environment and the host environment. Through which process it communicates through endocytosis and exocytosis. Through the two process, this parasite and host communicate. For that communication, this flagellar pocket, that invagination of the cell membrane of the masticore, stay as a site. So that's about the features of hemasticoid form. Then what about the infection? This hemasticoid form, it multiplies by the process of binary fission. Binary fission while staying inside the reticuloendothelial cell. Okay. And as it continuously multiplies and its number when become 50 to 200 parasites, then this parasites means that collection of parasites, it rupture the wall of this endothelial cells and comes out. Then it will, then that parasite in amasticoid stage infect another reticuloendothelial cell and repeats the same in the process. And in that way, its number increase in the body of the host. And a few parasites remain in the circulatory system of the infected person. Okay. Next, we are going to see the promasticoid stage or leptomonad form. Its picture is shown here. It is the flagellar form of the parasite. How that amasticoid form get transformed to promasticoid form when When a sandfly bite an infected Persian that carries the hematocyte form, that hematocyte form enters the gut of the sandfly. After getting inside the gut of the sandfly, hematocyte form get transformed to promastocyte form. Okay, then what are the features of this one? This promastocyte form. It is long spindle shaped form and its length is 15 to 20 micrometer. Breadth 1 to 2 micrometer. At the anterior end, we can see the kinetoplast and a nucleus at the center of this body. And we can see a flagellum, single flagellum protrude outside the parasitic body that originate from the kinetoplast or basal body. And that flagellum can be longer than the size of this promastocyte, or sometimes its length be same as that of the length of this promastocyte stage. That will vary. Then how it multiplicate? It also multiplicate through binary fission. And as the parasitic population increase in the gut of sandfly, that infection, that number of that parasite, it moves to the it accommodates the pharynx and later the proboscis of that sandfly. By the ninth day of parasitic infection, that parasite completely blocked the entrance, buccal cavity, pharynx, and proboscis of the sandfly. That means that is soaked with infection. That number of parasites in the body of sandfly be abundant at that stage. And then at that stage, when this sandfly bites a healthy human being, then the pro-masticoid form of parasite from the sandfly enters the body of new host and inverts its the reticuloendothelial system and transform to a masticoid. Clear? So, from sandfly, promastocyte form enters the host and transform to a masticoid form. Then mode of transmission, how it transmitted. We know that simply it is transmitted through the bite of sandfly to a healthy human. There are 21 to 30 different species of parasite is there that can infect humans. And also the sandfly are also of different species. They are Mediterranean vector, Indian vector, Sudanese vector. Likewise, so many different varieties of sandflies also exist that can act as a vector of this parasite. Then method of transmission, how that infection is transmitted. The basic method is that through the bite of the sandfly uh, to a healthy man to suck his blood. By that time, that parasite enters the body of that healthy man and reaches his blood circulation. That's the basic method. And the second one is through coitus. Coitus means sexual relationship between a male and female. And during that time, if one partner is infected, there is chance for the transmission. And also, while if mother is infected, then the child in her uterus, there is chance for or receiving that infection. And the last one is through blood transfusion, by transferring blood. If that donor is infected, then there is chance for having the infection. Along with the sandfly, there are so many other vectors that can carry this parasite. They are dogs, rodents, etc. Okay. In the different types of transmissions, it is special to mention about anthropootic transmission. Anthropootic transmission means here an infected human being is essential to maintain the transmission of parasitic cycle. In that case, we can control the disease, that spread of that infection through effective treatment of the infected patients. If the transmission is anthropogenic type. So there we know that clearly how we can control, because that infected person is the main carrier of that parasite. So by controlling or treating his disease, we can control or we can mitigate that infection. Then life cycle. Life cycle proceed through so many stages. First stage is the biting of sandfly to a new host for taking blood meal. And while that time, the promasticoid form of the parasite that present in the proboscis of the sandfly will enter the host body. Okay, and as the promasticoid form enters the body of host, it is phagocytosed by the phagocytic cells that present in the host. By that way, that parasite invade inside the phagocytic cells in the host body. Then after the phagocytosis, what will happen? That phagocytic cell, that parasitic, the promasticoid parasitic cell get transformed to a masticoid form. That a masticoid form is a tissue-specific stage of parasite. And then the masticoid form, it continues its multiplication inside the reticuloendothelial system and increases number. And in stage 5 and 6, as the infection spread, the parasite and many other factors in the host body, it decide whether to produce any symptoms or to affect any visceral organs in his body, or to produce the cutaneous leishmaniosis. Okay, that all things depends upon many factors in the host and the parasite. And at that infectious stage, when a phlebotomine sandfly bite that person, then from his body, the infected macrophages enters the body of sandfly. And in stage 7, that amasticoid form that enter the body of the sandfly from that infected person, it transforms into the masticoid form of the parasite. Again, then in the last step, stage 8, that transformed promasticoid form from the gut of the sandfly multiplies and again migrate to the proboscis to bite another healthy human being and to transfer the disease. Okay, I think that's clear to you. With this, I'm going to wind up today's section. Thank you.
I'm an icon, man. Ain't my girl about a 10. I should fuck her in a Benz.